Let's start with the basics. What are T-cell receptor therapies?
TCR Ts are created by engineering a patient's T cells to express a cancer-specific T cell receptor. And this enables T cells to recognize antigens that originate from proteins that are found inside cancer cells. So cells in our bodies naturally present these antigens on the cell surface that are derived from proteins that originate from inside the cell. And this enables T cells to survey cells throughout the body and recognize infected cells, but also potentially cancerous cells. So TCR Ts are really taking advantage of this natural mechanism of host defense.
So we're exploring a range of cell therapies. How are TCR Ts different?
So TCR Ts can identify intracellular antigens, and this enables us to unlock a much broader target landscape than modalities that only recognize antigens or proteins that are present. And the precision of our TCR therapies really comes from the fact that we're targeting cancer-specific antigens, and this therefore enables highly selective targeting.
How do you design the right TCRs for targeting tumors?
We've designed a range of complementary technologies that enable us to identify the right targets for TCR T, isolate cancer-specific T-cell receptors, and also optimize the functional properties of those TCRs. A high-throughput discovery TCR discovery platform uses a functional genetic screening technology that enables us to identify cancer-specific TCRs from a range of different sample sources. Furthermore, our TCR discovery platform is actually serving as a foundational technology to train AI tools that will inform the next generation of TCR Ts.
Okay, let's talk about targets. What are some of the most important TCR T targets today, and why are mutations in p53 and KRAS so compelling?
So most cancer mutation-derived neoantigens are unique to a given patient's cancer. However, there's a few that are actually derived from common oncogenic mutations in cancer-driving genes such as KRAS and TP53, and these are shared between patients and are referred to as shared neoantigens. Now these shared neoantigens, because they're highly recurrent across different patients with a range of different cancers, means that they're very attractive targets for TCR T therapy. And we have a number of early-stage clinical TCR T programs targeting driver mutations in KRAS and TP53 that are being investigated in a range of advanced solid cancers, including pancreatic, colorectal, and breast cancer.
So, solid tumours have long been challenging to treat with cell therapy due to the high tumour heterogeneity and the immunosuppressive tumour microenvironment. What are we doing to address this?
So firstly, we're targeting cancer mutations that actually play a key role in cancer development and progression. And secondly, much like with our CAR-Ts, we're developing armoring strategies that are designed to optimize the activity of our TCR-Ts in solid tumors. So we're knocking out genes such as TGF-beta receptor 2, which is designed to enable our TCR-Ts to resist immunosuppressive signals that are present within the tumor microenvironment. We're editing our T cells to potentially enhance their function by knocking out genes such as CBL-B, which is a known negative regulator of T cells. And finally, we're also editing our T cells with the aim of improving the activity of both cytotoxic and helper T cells via the knock-in of the genes encoding the CD8 co-receptor. So Mark, today AI is everywhere. It touches most areas of our lives. How do you see AI in helping us move faster or more precisely in developing TCR T-cell therapies?
Well, you're absolutely right, Gavin. AI is integral to our R&D strategy, and it's important in terms of how we improve the drug discovery process. We're using AI-powered tools to advance screening and target identification. For example, AI can help us analyze complex tumor immune data much more accurately than we could do, and much faster than we could do before, and prioritize potential TCR T targets and identify optimal T-cell receptors.
So zooming out, what is AstraZeneca's broader strategy for cell therapy, and how does TCR T fit into that?
Yeah, I think what we're doing is we're trying to attack tumours in a number of different ways, and so we've got our CAR T portfolio and chimeric antigen receptors showing promise in a range of different cancers at the moment, and we have a TCR T portfolio. I think attacking the tumours from different directions and using different technologies is the best way in which we can advance here. We're already exploring technologies such as armoring. We're exploring other ways in which cells can be controlled better and perhaps expanded inside the patients, and really identifying ways in which we can deliver cells. We're looking at off-the-shelf therapies, in vivo CAR Ts.
So finally, Mark, looking ahead, what does the future look like for TCR T?
Well, the aim is really we want to deploy these therapies. So I think what we've learned in this space is that the earlier you give these treatments, the better the outcome. And we also know that patients with reduced tumour burden tend to do better. So we know that the cancer therapeutics are often being given in combination. And so what other medicines can be combined with cellular therapies to really sort of increase and maximize the benefit those patients derive from them. I think the future is very bright in this space, and we've still got a long way to go. Well, thank you, Gavin, for your time. This has been absolutely excellent.
Thanks, Mark. Great to talk.